Antimicrobial resistance happens when germs like bacteria and fungi develop the ability to defeat the drugs designed to kill them. Resistant infections can be difficult and sometimes impossible to treat. Antimicrobial resistance is a naturally occurring process. However, increases in antimicrobial resistance are driven by a combination of germs exposed to antibiotics and antifungals, and the spread of those germs and their resistance mechanisms. Definition of germs and antimicrobials. Antimicrobial resistance does not mean our body is resistant to antibiotics or antifungals. Rather, it indicates that the bacteria or fungi causing the infection are resistant to the antibiotic or antifungal treatment. Germs are microbes which are very small living organisms including bacteria, fungi, parasites, and viruses. While most germs are benign or even beneficial to humans, certain strains can lead to infections, with these harmful germs termed as pathogens. Antimicrobials refer to describe drugs that are designed to treat many types of infections by either killing or slowing the growth of pathogens responsible for the particular infection. There are also antivirals or antiparasitic resistant. Bacteria cause infections like strep throat, foodborne illnesses, and other serious infections. Antibiotics are effective in treating bacterial infections. Fungi, on the other hand, cause infections such as athlete's foot, yeast infections, and other significant ailments, and antifungals serve as the primary treatment for fungal infections. It's common for people to use the terms antibiotic and antimicrobial interchangeably. The impact of antibiotic and antifungal use on resistance. While antibiotics and antifungals play a crucial role in preserving lives, their usage While antibiotics and antifungals effectively eliminate certain germs responsible for infections, they also eradicate beneficial germs that shield our bodies from infections. The germs that survive from the effects of antimicrobial drugs gain resistance and proliferate. These resilient germs harbor traits of resistance in their DNA, which can subsequently disseminate to other microorganisms. Resistance mechanisms and spread of germs. In order to survive, germs can develop defense strategies against antibiotics and antifungals, resulting in infections that are difficult or impossible to treat. DNA tells the germ how to make specific proteins, which determine the germ's resistance mechanisms. Bacteria and fungi can carry genes for many types of resistance. When challenging to treat germs possess the right combination of resistance mechanisms, it renders all antibiotics or antifungals ineffective, resulting in infections that are difficult or impossible to treat. Alarmingly, antimicrobial resistant germs can transmit their resistance mechanisms with other germs that have not been exposed to antibiotics or antifungals. Some of the few examples of defense strategies used to resist the effects of antibiotics or antifungals are: 1. Limit antibiotic access. Germs employ strategies to restrict antibiotic access by altering entry points or reducing their number. For instance, gram-negative bacteria contains an outer membrane that serves as a protective barrier from the environment, selectively preventing antibiotic drugs from entering. 2. Eliminate antibiotics or antifungals. Germs. Eliminate antibiotics through cell wall pumps that expel the antibiotic drugs entering the cell. For example, certain Pseudomonas aeruginosa bacteria produce pumps capable of removing various crucial antibiotic drugs, including fluoroquinolones, beta-lactams, chloramphenicol, and trimethoprim. Similarly, some Candida species use pumps to eliminate azoles like fluconazole. 3. Modify or degrade the antibiotic 4. Alter antibiotic or antifungal targets. Many antibiotic drugs are designed to target specific components of bacteria. Germs modify the antibiotic's target, making it incompatible and ineffective. For instance, Escherichia coli bacteria carrying the MCR1 gene can modify the cell wall exterior, preventing the drug colistin from binding. Another example is Aspergillus fumigatus, altering the CyP1A gene, preventing triazoles from binding to the protein. Lastly, 5. Bypass antibiotic effects. Germs develop new cell processes that avoid using the antibiotic's target. Example of this is that certain Staphylococcus aureus bacteria can bypass the drug effects of trimethoprim, showcasing their ability to adapt and survive in the presence of antibiotics.